Hi, my name is Neil Frick. I'm Senior Vice President for Medical Programs and Information at the National Bleeding Disorders Foundation. I'd like to welcome you all to this recorded version of Gene Therapy, Real-World Experiences and Current Challenges. It was a medical track session which took place live on August 18th, 2023, during the National Bleeding Disorders Foundation's recent annual conference in National Harbor, Maryland. I have the distinct pleasure of introducing Dr. Glenn Pierce. Dr. Pierce is currently an independent consultant and serves as the Vice President of Medical Affairs at the World Federation of Hemophilia and serves as a member of the WFH USA Board of Directors and the National Bleeding Disorders Foundation Medical and Scientific Advisory Council. Dr. Pierce has 30 years experience in biotechnology research and development from the bench to the bedside in small, large, public, and private biotech, biopharma firms, including Biogen, Bayer, Inspiration, Avogen, Select Genetics, and Amgen, in the areas of tissue regeneration and hematology. He is the co-author of more than 150 scientific papers and has been awarded over 15 patents. Dr. Pierce's talk is entitled "The Science Required to Cure Hemophilia." Dr. Pierce.
Thank you, Neil. I appreciate the introduction. Good day to everyone. I am going to talk about the science required to cure hemophilia in this talk. My disclosures: I work with a number of companies that are working on gene therapy, both in the hemophilia space and outside of that space. So let's look at the history of the last 60 years and the remarkable technological changes that have occurred with regard to hemophilia treatment. We started those 60 years with plasma-derived clotting factor concentrates, which unfortunately, although they were very effective in stopping bleeding, led to widespread viral contamination with HIV and hepatitis viruses. That accelerated the development of recombinant clotting factors. In the 1990s, these were approved and really entered us into a new world of protein engineering. Can we make better proteins through recombinant DNA technology? And that was developed over the next 20 years or so with the synthesis of extended half-life clotting factors, both factor VIII and factor IX. And those extended half-life clotting factors improved treatment as well, and eventually led to the development of an ultra-long half-life factor VIII called efanesoctocog alpha. We also saw the development of antibody therapy, antibodies that could mimic factor VIII and replace that function that accelerates the development of a thrombin burst. And that was an exciting development beginning in 2017. And now we're in an era where gene therapy has been approved, one each for factor VIII and factor IX, and we have in trials rebalancing agents, as well as new factor VIII mimetics and new gene therapies. What's most important about these technological developments, though, is what they do for treatment of hemophilia, and they have allowed us to go from on-demand therapy to treat bleeds that have already started to literally 24/7 coverage without peaks, without troughs, that allow for a more constant level of a factor VIII or a factor IX activity that can provide for protection from bleeding, and eventually to a cure. Now, when I talk about cure, I really want to make sure we keep our eye on the ball, because curative therapy is the ultimate goal. And 24/7 coverage of with some of these newer technologies is heading in the right direction. The dream of every newly diagnosed family is a cure, and the dream of every affected person is a cure. And it's been out of reach until now for some. We've been promised the cure since the day I was born, practically, and we're finally now seeing the realization of that. I'd also like to see if we can agree on the definition of a cure. We probably can't, but my definition would be free of bleeding and use of any coagulant therapies for a prolonged period of time, measured in years. Has it been achieved? Well, it's definitely been achieved with liver transplant. that does affect a permanent cure of hemophilia when one receives a liver transplant usually for end-stage liver disease. And then for some gene therapy recipients, possibly they are cured. And it remains to be determined with time, but it looks promising. Technically, these are all functional cures, since the affected person can still pass the variant gene, the hemophilia gene, on to offspring. Now, let's speculate for a moment and broaden the definition of a cure and just begin to imagine what a cure could look like if we started using maybe some different definitions. Would an injection every 5 years, every 1 year, every 3 months, or once a week qualify if the person was bleed-free during those intervals? Non-viral gene therapies, RNA therapies may offer periodic injection or topping off of the factor levels in the future. Antibody therapies may allow for that too, future antibody therapies. And then gene editing platforms, where a corrected gene is actually inserted directly into our chromosomes, for instance, in the liver, and they would offer the closest curative alignment to a liver transplant. So if we imagine a little bit, broadening that definition, we can see where research can take us in a number of different directions that may be of benefit to develop curative therapies for hemophilia. Now, I also want to comment on the current therapies that are being used because they're primarily using adeno-associated viruses, which we'll hear more about in this session, AAV, and those have been around and used for gene therapy for the past 30 years or so. They've had a number of false starts along the way, hurdles that have come up, problems that have had to be solved. To the point now where we now have several products that are approved in a number of different locations, geographies around the world that use AAV, including one each for hemophilia A and hemophilia B. But this is a useful framework on this slide to try to understand what the risks are for AAV gene therapy. And we can break it up into what we know, and we know that there are occasional peri-injection reactions that need to be managed in the hospital when the gene therapy is given. We know that proportion of patients develop increases in their liver enzymes, and that those can be treated with steroids, but then if the steroids are given in too high of a dose or for too long a period of time, there are steroid-related toxicities. We also know what we don't know. We don't know what the significance of these liver enzyme increases are in year 1. And importantly, in the AAV5 factor 8 phase 3 trial, we don't know the significance of liver enzyme increases in over a quarter of the patients in years 2 and 3 following therapy. And then we also know that some of the AAV does get integrated into the chromosomes, and we don't know what the consequences of that. What about what we forgot we know? Well, we forgot we know that AAV does integrate, and much of the field continues to believe it does not, but it does. It integrates into the host genome and has unknown consequence. And we also know that AAV in other diseases can cause acute liver failure. At doses only slightly higher than the doses used in hemophilia, children have died from acute liver failure as a result of the AAV host immune response. And then the most problematic area is what we don't know. And of course, we can't know what that is at this point, but we can go back 4 or 5 years and realize that we had no idea that in other clinical trials with some other AAV serotypes or cousins of AAV5 that are used in hemophilia, we have seen thrombotic microangiopathy, complement activation, dorsal root ganglion toxicity, and hemophagocytic lymphohistiocytosis. And so we don't know what the future holds, but we were surprised by all of these adverse events, and therefore it's reasonable to assume that there are more unexpected events that may come along with this new technology, this new therapy. So where are we in 2023? Do we have a once and done cure for hemophilia? Well, maybe we do, at least for some patients with hemophilia B, but maybe not for hemophilia A. So let's look at all the considerations. Ability to retreat? Well, not with AAV. We develop very large antibody responses that preclude giving the AAV again. For Hem B, it may not be needed. For Hem A, we can't do it. Ability to boost a low result? No for both. Predictability of the need for steroids or other immunosuppressive drugs? No, but it appears to be short-term needs in Hem B, and there are some long-term needs that go out over a year for Hem A. Probability of an acute infusion reaction? No to both. Knowledge of the safety unknowns that I just referred to? No to both. Risk-benefit analysis favorable in high-income countries? Perhaps yes for Hem B. Hem A has many alternative therapies that one needs to consider in a shared decision-making process. Risk-benefit analysis favorable? Well, in lower-income countries, that answer is probably yes for Hem B, and it may be for Hem A if one is looking for a short-term respite from bleeding with their hemophilia. And then works in children? No to both. So we do need shared and informed decision-making. It's essential as one looks at these kinds of new therapies. But I want to point out that the informed decision-making is really only as good as the available data. And we don't know a lot, as I showed you on the previous slide. And so we have to be careful when we talk about informed decision-making. Now, it's important for us to be able to identify rare signals like the kind I've showed in rare disease populations that are really spread out geographically. And it's important to have a registry. And the World Federation of Hemophilia has developed a gene therapy registry where we would like to see all patients who receive gene therapy go into this for a prolonged period of time to look at long-term safety, durability, variability, and efficacy. And I would point out for all of us, patient safety is our responsibility, especially in dealing with technologies where we don't have many answers. So I'm going to now talk about some solutions, and I'm going to divide that into solutions to deal with the problems of using AAV to carry genes into cells. I'm going to talk about non-AAV solutions, and I'm going to talk about the factor VIII transgene itself, seeing if we can improve that because it is problematic and causes cell toxicity when it's inserted into cells. And then the factor IX transgene, I'm not going to talk about that. The field is using Padua, which is a single amino acid mutation that increases the specific activity of factor IX 7 to 8-fold and really has enabled therapeutic factor IX gene delivery. So to start off with AAV, as we can see in this cartoon, enters the cell. We have no idea how, but once it's in the cell, it goes through a whole series of processes that I won't go into a lot of detail for. And eventually, protein comes out the other end, and that protein circulates. But this is essentially a black box. We have no idea quantitatively what the AAV does, how it gets from the endosomes, where most of it is actually digested and destroyed, where a small amount gets into the nucleus, uncoats through unknown mechanisms, forms single-stranded DNA that moves on to double-stranded DNA, forms stable episomes and concatemers, and then is able to synthesize RNA or transcribe RNA that then leaves the nucleus, goes into the cytoplasm, makes protein that goes through the endoplasmic reticulum and Golgi. And it's in this whole series of steps that we don't fully understand. In fact, we have very little understanding of it. We don't know what controls it. We don't know the enzymes that control the creation of the DNA in a stable form or anything like that, even though we've been working on this virus for the past 40 years. So that's a problem if we are looking to develop this as a therapy. I also want to point out that we need to put in to the human being over 100,000 AAVs per hepatocyte in order to see the kind of therapeutic effect we have. So this is an extraordinarily inefficient process. And those 100,000 AAVs cause quite an immune response, or a series of immune responses. So if we can make any improvements on this, decreasing this ratio, that would certainly improve the immune response or decrease the immune response that the host makes. So let's talk about the variability of the responses we see to AAV therapy. I'm going to first speak about manufacturing, and then I'm going to talk about us, because we are the human hosts that manufacture the AAV. Our livers are 1.5-liter bioreactors. The variability that we see is not different than the variability we saw when recombinant proteins first started getting made back in the early 1980s, late 70s, early 80s. We didn't really know how to do this, so it was an iterative process, a learning process, and was very challenging. It was very challenging to make the initial factor VIII recombinant proteins. But eventually, this became like a cookbook and a well-worn recipe that could be reproduced faithfully every time. That's where we need to go with AAV. So what causes the variability? Well, there's two sources. One is the actual manufacturing of the AAV vector, the upstream production in big stainless steel bioreactors to make the AAVs, and then the downstream purification to purify the vector genomes, the actual AAVs that contain the factor VIII or the factor IX molecule. And then we also need quality control assays, identity assays, to be able to determine if we're making the same batch every single time. And those assays I'll speak to on the following slide, but they're not very good. The second problem we have that's very difficult to deal with is that we are human bioreactors. So we're not talking about 20,000 liters stainless steel fermenters where we can control every variable. As occurs with recombinant protein production. We're talking about us, and we all have different enzyme systems within our bodies, within our livers. We all have different pathways for the AAV to get to the target cells. And so in each of us, we're assuming that we all work fundamentally the same way to get the AAV into the cell and get protein secreted. But the variability is present within each of us, and that's difficult to challenge. So those are the two sources of variability that are difficult to deal with, that we see with AAV gene therapy. So when we look at manufacturing, on the left-hand side are many of the tests that are used to ensure that each batch is similar to the previous batch. But all of these tests are really only as good as the assays. And sometimes the assays aren't very good. And so there is batch-to-batch variability in producing AAVs. And that's been demonstrated with products that are going into the clinic. So it's the best we can do at this point in time, but it shouldn't be tomorrow's state of the art. A big problem here is that there's not a lot of disclosure about the assays. The companies consider this to be proprietary, and so patients as well as healthcare providers don't really hear anything at all about these assays. And that's a problem. It's a problem because the assays sometimes don't work very well. A number of us are concerned that there's not sufficient research going into the development of AAV therapeutics to deal with the lack of predictability, the variability, especially with regard to factor VIII. And so we are calling upon the manufacturers, the companies, as well as the FDA to develop more rigorous testing for being able to recognize that the assays are not entirely what we need, and more research is needed in the manufacturing process for AAV. I want to turn now to factor VIII transgene modifications, which I said I would mention before. Unfortunately, we don't have a factor IX Padua variant for factor VIII, but biochemists over the last 30 plus years have dissected the factor VIII molecule, made a number of different kinds of point mutations, identified variants, and there have been variants identified that have increased secretion that might be potentially useful at decreasing some of the burden that cells have in making the native factor VIII or the B domain deleted factor VIII. And so this is an area that needs to be of intense interest going forward to see if we can't decrease some of the variability and some of the toxicity associated with factor VIII, AAV factor VIII gene therapy. I want to turn now to non-AAV gene delivery, and the holy grail is to use a non-viral approach, but that has proved to be very difficult over the years, although continued scientific progress is being made. And as you all know, we can now deliver RNA with lipid nanoparticles very effectively, such as the COVID vaccines. But the RNA only needs to get into the cytoplasm. It doesn't need to get into the nucleus. So we need an LNP that can get into the nucleus. One approach is using transposons. Transposons are large pieces of DNA that can contain our DNA cargo, such as the factor VIII gene. And there are these enzymes called transposases that can break open the DNA at various points in the chromosomes and allow for the insertion of the transposon. And so this is an approach that is non-viral and can offer potentially, at least in mice, a therapeutic dose. It's also, since it doesn't cause an immune response, able to be redosed, and that's what's shown here. 1 dose up to 4 doses increases the factor VIII level in the mice. But what the transposons do is insert into the DNA at random, and that's potentially problematic because they could insert into an area of oncogenic potential. Most of the insertions are in introns and intergenic regions, but some do go into the genes themselves, the exons. So I want to turn to something that might be more specific now, and that's gene editing. And gene editing is a type of gene therapy, and it can either be used to knock out a gene, such as transthyretin or PCSK9 to decrease cholesterol, or it can be used to repair a gene, to correct genes with a few mutations. That's not a likely scenario for us with factor IX or factor VIII because there are so many different mutations that cause heme A and heme B. Or it can be used to insert, and with an insertion, it can put our gene of interest into a safe harbor locus. So lots of approaches with gene editing that's only really been recognized as something that's relatively easy to do for the past 10 years or so. I want to talk about two approaches for gene editing. One is with Regeneron and Intellia, 2 biotech companies on the East Coast of the United States, and they're working with factor IX. And what they're looking to do is to use gene editing to put the factor IX into the albumin locus and have it get driven off the albumin promoter, which is shown here. And so this is something where the lipid nanoparticle delivers the nuclease enzymes, the Cas9, and the guide complex, and then an AAV is delivering the factor IX. So we're still dealing with AAV, but in this case, the AAV doesn't have a promoter, and just a sufficient amount needs to get in that the enzyme can work on to be able to integrate into the albumin promoter. So this is something that is moving toward the clinic for factor IX deficiency. Another approach is to use another cell type because the Regeneron approach is going into the liver. And so another company is working on engineering B lymphocyte medicines. And using genome engineering to take B lymphocytes and insert a new gene, such as a factor IX gene, into those. And the company has been able to show in mice that it goes into the bone marrow of the body. So you can see it in all the bones. It's also in the spleen and in the liver in this early time point as well. And so what we can see is that the B lymphocytes are very much like hepatocytes. They're protein-producing cells. They make all of our antibodies. And so we can take a B lymphocyte and engineer it, and it should have adequate protein machinery to be able to make the factor IX molecule. And so the engraftment occurs without preconditioning either, which is different from the work that's been done with lentiviruses that require toxic preconditioning to put back into the bone marrow. So very simply, B cells are isolated from a patient. They're engineered. A new gene is added with gene editing. And then they're growing up, expanded, and differentiated into a terminally differentiated B cell called a plasma cell. And then they're just infused back in. And that's where they home into the bone marrow and produce the factor IX. So this has been done in primates at this point, and so this is very encouraging. And I'd like to turn to another type of gene editing where they're working on finding different nucleases that might offer better specificity and better efficiency of insertion into the host chromosomes. And so this is a company that is pursuing new Cas9, new Cas nucleases and different CRISPR systems in a variety of different places, places where we really haven't encountered them, and so we wouldn't necessarily have antibodies. These all come from bacteria, and so they've gone into hostile environments where humans don't go, such as undersea hot vents or hot springs, hot vents, and look for undiscovered nuclease enzymes. And what they have been able to find is that they can find nucleases that have higher specificity, less off-target activity, which is important as one contemplates developing this for human therapeutics. And what they've shown with some of their proprietary Cas nucleases is that they can easily knock down the protein, get 90% knockdown, and be able to eliminate that gene from being able to make protein. And so really the next step is being able to do editing and insert a gene into the area that has been disrupted. And that's what they're working on at this point. So I have a few final thoughts. I'd say it's fair to say that first generation gene therapy is here. Factor IX appears to be more reproducible and more durable than factor VIII, an important distinction. Future iterations of gene therapy will include the different gene editing approaches I've talked about, non-viral delivery of DNA and RNA, and the development of new factor VIII transgene variants. Better factor VIIIs, the way we have a bio-better factor IX in Padua. The technology for gene therapy is moving very fast. By the time one gets through the clinic, new technologies have already come along. And so it's important for us to stay very close to this, understand what tomorrow's discoveries might be, and how tomorrow's discoveries would make gene therapy safer, better, and cheaper. And that's important as we think about the entire world, including the 85% that don't have access to therapy. Hem A does need a permanent cure. Both Hem A and Hem B children need a solution, and that solution is unlikely to be AAV. So we need something different for kids. Safe and effective gene therapy for lower income countries, as I mentioned, it represents the only long-term viable solution to address this huge health inequity that we have largely ignored over these past 60 years as the pharmaceutical industry has focused on the 15% who can pay high prices for these medicines. I thank you for your attention. Good day.
On behalf of the National Bleeding Disorders Foundation, I want to thank Dr. Pierce for recording this excellent presentation today. Thank you, Dr. Pierce.
You're welcome.